Hello, everyone. Thank you for joining this new but also last session of the BioClock Academy lecture series. We will have a summer break from June to August and start freshly in September. Happy to see many familiar but also new faces here in the virtual space. My name is Wana Georgiana Roos Oswald, and I'm a postdoctoral researcher in the Dutch BioClock Consortium. And together with Hannah Brooks, who is a PhD student in the same consortium, we host the session today. The BioClock Consortium is a Dutch research organization, and it is set up to study different aspects of the biological clock, so in humans, in health, and in society. And the BioClock Academy is a lecture series organized within this consortium, and it is addressed actually to everyone who is interested in the field of chronobiology, but especially addressed to early career researchers. So its aim is to introduce you to the basic concepts, equip you with the proper knowledge by experts in the field, and it takes place every third Wednesday of the month. With respect to the time frame, we will have a 40 minutes talk followed by 15 minutes of discussion roughly, so that we can finish in 1 hour. So we will finish for Amsterdam time around 5 p.m. But for the speaker today, it's early morning. So thank you for being so early here. So maybe just in time to have an early breakfast afterwards, the well-deserved one. During the talk, you can also write your questions in the chat and we can discuss them afterwards. But you can also raise your hands afterwards and we have a proper discussion. And now I give the floor to Hannah, who will introduce you to our guest speaker today.
Yes, hello. So also a warm welcome from me. And indeed, I have the honor to introduce you to the speaker of today, Professor Susan Golden. She's the director at the Center of Circadian Biology at the University of California, San Diego. And Susan will bring us a very nice new perspective to this academy series, the one of circadian clocks in prokaryotes. So I think that's very interesting. So in the early 1990s, Susan actually began a collaborative project that demonstrated the circadian rhythmicity in gene expression in the cyanobacteria Synechococcus elongatus. And this is now the model organism to study circadian clocks in prokaryotes. And this is also the model system where Susan and her colleagues in the lab work on to explore and investigate how the circadian clock functions at a molecular level as a timekeeping mechanism, and then how it is integrated more at the cellular level to control gene expression and metabolism. So yeah, I'm very much looking forward to this talk. So I would say, Susan, the floor is yours.
Wait, thank you very much. I'm really pleased to be here. I know about the Dutch BioClock Consortium. I'm actually served as an external advisor for the group, and I'm delighted to be here, even though it is 7 a.m. in San Diego. So I'm going to be telling you, as you heard, about circadian regulation in a prokaryote, in a cyanobacterium. And I've also been asked to tell you a little bit about not only about the research, but about how I got to where I am today and how this work evolved. And in particular, I have to really credit that I am here talking to you today because of, because of two very exceptional collaborations. And I'll be telling you more about these collaborations, but one that began in the 1990s with Takao Kondo, Masahiro Ishiura, and Carl Johnson. So a Japanese Vanderbilt, and at that time, Texas, where I was, consortium, and then a newer collaboration with Andy Parch, with Andy Lee Wong and Carrie Parch in all of us in California. But the way that I got here was not really expected, because I'm a cyanobacteriologist. I did not start my career at all working in circadian rhythms, or even with an organism that anyone at that time expected to have circadian rhythms. So I want to start out a little bit telling you what cyanobacteria are, because I don't expect that most of you know. These are photosynthetic prokaryotic organisms. You are probably familiar with them, even if you don't know that you are, because they are literally everywhere, including at a Manhattan nutrition store where they're called blue-green algae. And you can see that they are beautiful and diverse. And I'd like to talk a little bit about that diversity and give you an idea of how important these organisms are. So first of all, you may not realize that cyanobacteria were responsible for the single most cataclysmic event that's happened since life evolved on Earth, and that is that they formed the oxygen atmosphere of Earth. And because we need and thrive on oxygen, we don't think about how corrosive and damaging this molecule is. So in fact, there was life on Earth when cyanobacteria arose, and they killed pretty much all of it off. Changing our atmosphere to contain oxygen is something that everything on Earth has had to evolve to be able to adapt to. The pictures that you're seeing here are actually fossil cyanobacteria. The oldest known fossils, living fossils, are in fact from cyanobacteria. In addition to undergoing a plant-type photosynthesis that evolves oxygen, they also are among the a few groups of organisms that can take nitrogen out of the air and fix it into nitrate and ammonia that can be used by living organisms. So they're very important metabolically. And they're also very diverse. So this first slide, which shows some morphological diversity, but first sets out to dispel any idea that you may have as a eukaryotic researcher that bacteria are single-celled organisms. There are single-celled organisms, but there are many, many multicellular bacteria, some of which even differentiate specialized cells, as you can see here, and have essentially have tissues and are much more complex than were thought many years ago. You are maybe familiar with them because cyanobacteria can form harmful algal blooms. I know this has been a problem in the Netherlands in some lakes. And also, if you drink a green energy drink, there's a good chance that one of these cyanobacteria, the curly one that you see here, spirulina, is probably in that drink. They're also in every habitat on the planet. So they thrive in the open ocean. They are present in the cold deserts of the Arctic and Antarctica. They're present in the hot deserts, where they're important for establishing the desert crusts that keep all of the sand from blowing away. And many of them also live in symbiosis with eukaryotic organisms. So cyanobacteria also, so having evolved on the Earth for almost longer than anything else that we have now, shares with eukaryotic organisms, with humans, with plants, with fungi, other kinds of animals, also shares having circadian rhythms. And I don't need to tell you what circadian rhythms are, but these properties that are shared by different eukaryotic organisms, I just want to assure you the circadian clock of cyanobacteria actually fulfills all of these criteria. They are genuine, bona fide, genetically programmed 24-hour periodicities that are synchronized with the local environment and are temperature compensated. So I want to say something about how circadian rhythms were found in cyanobacteria, because it wasn't by looking for them. And I should say that there were biases in the field prior to the work that I got involved in. There were biases that bacteria couldn't have circadian clocks because because there was an expectation that the cells were just too simple to support a clock. They didn't have a nucleus, and that sometimes their lifetime is less than a day, and it just kind of didn't make sense. So people weren't looking for rhythms in cyanobacteria. What happened, though, was there were cyanobacteriologists who were trying to explain a conundrum. And this conundrum is, how is it that there are some single-celled cyanobacteria that can undergo photosynthesis and give off oxygen, and they can fix nitrogen out of the environment. And the reason that this was a conundrum is that the nitrogenase enzyme is notoriously oxygen sensitive. And I emphasize single-celled cyanobacteria because there are multicellular cyanobacteria that solve this problem spatially. They they differentiate specialized cells to fix nitrogen. These undergo processes that keep the oxygen environment inside very low. And then you have vegetative cells that are undergoing photosynthesis, fixing carbon and giving off oxygen. And then you have the heterocysts that are fixing nitrogen. But a single cell doesn't have that luxury. And so what was discovered was that, in fact, the separation of these processes is done temporally. And this work was done not by my lab at all. It was done prior to me getting into the area of circadian rhythms. And it kind of evolved in fits and starts, because there were really a number of reports in the 1980s, some even in the late 1970s, that showed these odd rhythms, but the people working on cyanobacteria didn't know what to think of them. And the concept of what people were studying in plants and animals that they called circadian rhythms were not, that knowledge wasn't really spread throughout the field. But one of the first reports that looks really very convincing that you'll see here was with a unicellular cyanobacterium in a lab of a scientist named Mitsui in Florida, who found that when cells were put in a light-dark cycle, there was an oscillation of photosynthesis that you see here, peaking in the daytime, and in the dark, then nitrogen fixation. And what you can see is that when the cells were kept in constant light, these rhythms persisted. However, Mitsui did not conclude that these were circadian rhythms. He was growing the cells under conditions where they were dividing every 24 hours, and he concluded that he was looking at processes that happen at different phases of the cell cycle, and that for that reason they were separated. The actual discovery that cyanobacteria have a clock was done by a group, T.C. Wang in Taiwan, who saw work like Mitsui's and also did his own work on nitrogen fixation and photosynthesis in a single-celled cyanobacterium. But he, in fact, did say, oh, these rhythms seem similar to what people working in eukaryotes call circadian rhythms. Let me see what the criteria are for determining if it's a circadian rhythm. And in fact, he showed that amino acid uptake in the species that he was working with fulfilled all these criteria. Persistence in constant conditions, which hopefully will show up for you here. This is light dark, and this is continuous light. It could be reset by environmental signals, and it was temperature compensated. So these data made it really clear that cyanobacteria have a real clock. However, the organisms that had been studied up to this point were not genetic models. The people doing the work were not geneticists. And so the question is, how do you find out what that clock is? Well, as I said, I'm a cyanobacteriologist, and I started my PhD at a time that this species that we now call Synechococcus elongatus had just been recently discovered and found to be transformable. This is a unicellular freshwater cyanobacterium. It has a genome that's smaller than that of E. coli, only about 2,700 genes. And nicely, it takes up foreign DNA naturally. So it's very easy to transform. It's easy to knock genes out. It's easy to add genes in. And my PhD was focused on developing this organism as a genetic model. And the goal of developing it as a genetic model, though, was to study photosynthesis. So before I started working on the clock, I was working on photosynthesis. And I should say that I should remind everybody of the time frame here, which is we're talking about the 1980s. And so one thing I guess to emphasize is that all of the technologies that all of you know of whole genome sequences and bioinformatics, none of those things existed at that time. And what might surprise you, and actually it even surprised me at that time when I was taking biochemistry classes and I was learning photosynthesis and the Z scheme, is that the actual proteins that were responsible for the process weren't known. And so my goal was to develop a genetic model that would contribute to understanding what the proteins were, physically, biochemically, what is responsible for photosynthesis. And so my two of my early papers were on working out the conditions to transform this organism, which its old name was Anacystis nigilans that you see here, and looking at the expression of a family of genes that encode an important protein for photosystem 2. So this is showing you now what photosystem 2 looks like in either a chloroplast or a cyanobacterium. They're really quite homologous. And there's this very key protein called D1, and that's encoded by a gene called PSBA. And what I was, so at the time I was a graduate student, PSBA had been identified as an important protein in photosystem 2, but it was only while I was a PhD student and a postdoc that its role in being this protein was really discovered. And I contributed a little bit to that at that time. But what I then did during during my postdoc was to clone these genes, because it turned out that the organism had 3. And I should say that having paralogs in bacteria at that time was also pretty novel. And again, no whole genome sequences, so all of the genes were cloned the hard way, and studying what those genes do then was also much more difficult than it would be with modern technologies. But what I started my career on, my early independent career, was looking at how these genes are regulated, with the idea that if they all code for this protein, there's probably some reason there are 3. And I was looking at differential regulation. So what you can see is that one of the genes, when the cells are grown at low light, is expressed very highly. And then when you go to high light, the expression of that gene goes down and expression of the other 2 comes up. And then this last one is just to show that it's light and not electron transport, because an electron transport inhibitor was added. So this is what I was doing at the time that I got into the circadian field, because to try to study this regulation, we had just, we really wanted to do genetics to find out what the regulators were. And a particular genetic tool had just been developed and was being used a little bit in bacteria and a little bit in plants, and that was luciferase. So what my lab did to try to have a reporter was to take this gene and to make a luciferase reporter. And at that time then, I was contacted by Carl Johnson at Vanderbilt, who worked on a Chlamydomonas, a unicellular alga, and Takao Kondo, who was working on duckweed, lemna. And and Takao was doing a sabbatical in the US, and he was visiting Carl's lab. And they were talking about the fact that they'd rather have an easier genetic model. What's even simpler than duckweed or clammy, and what has a good genetic model? And after asking around a bit, they ended up landing on me and contacting me to see if I was interested in checking to see if my organism has a circadian rhythm. Because I'll remind you that at this time, there were good reports of rhythms in cyanobacteria, but they were not useful model organisms. I had at that time there were really only 2 good genetic models, and I was developing tools for it. And so Carl contacted me to see, to say, instead of trying hard to get these transformation in these other organisms, why don't we just assume that all cyanobacteria have circadian rhythms, and let's work with an easy one. And so that's what we tried. And so again, my organism is Synechococcus elongatus, and very easy to do genetics. And it turns out that it has very robust circadian rhythms of gene expression, which we were able to show using this luciferase reporter. So what you're looking at now are 2 plates of cyanobacteria that carry the same reporter, but the reporter is being driven by 2 different promoters, by one promoter that peaks at dawn and one promoter that peaks at dusk. So I think that what you can see is that these rhythms in gene expression are reflected very well by this luciferase reporter. This is an assay that is the type of assay you can do genetics with. So aside from the fact that the previous cyanobacteria weren't good genetic models, the rhythms that were being identified were very laborious, looking at amino acid uptake or nitrogen fixation, where you had to manually sample, and it really just wasn't practical to do big screens to try to find genes. But this kind of an assay, you can develop a genetic screen around. And just to show you how robust these rhythms really are, these are what the data in our first paper from this collaboration looked like. This is the reporter gene that I told you about. It's luciferase being driven by that PSBA1 promoter. And we could split a culture into half and have entrain one with 12 hours dark, 12 hours light, 12 hours light, 12 hours dark, and then start our monitoring in continuous light. And what you can see now is that the rhythm persists very robustly, and the cultures are 12 hours out of phase, having been entrained 12 hours out of phase. So very nice assay from a geneticist point of view. If you're going to do a genetic screen, this is what you want, something very robust. And also, we were able to show that this rhythm fulfilled the 3 properties you look for in a circadian rhythm, that we could give a dark pulse and we could reset the phase. And if you gave that pulse at different times, you would get different phases. So we were getting a phase response curve from it. And also that the rhythm is temperature compensated. So the Q10 is about 1.1, about a 10% difference in period over a 10-degree temperature range. Okay, so Takao Kondo set out to use this then use this assay to try to find mutants, because we really had no idea what we were looking for. So you're probably familiar with many clock genes, with cryptochrome, with period, in fungi, the frequency. At that time, only period and frequency were known, and we could not, and again, no genome sequences, but doing probing using those genes as probes, there was no indication that there was anything homologous. So we really had to screen to try to find the clock genes of the cyanobacterium. So what Takao Kondo developed was this turntable where mutagenized cells could be plated on these plates and get colonies, and those colonies then the plates would be positioned under a cooled CCD camera so that we could entrain everything to a light dark cycle, go into continuous light, and then about once every half hour, have this turntable turn so that a plate would be positioned under a cooled CCD camera, and the light generated from all of those colonies would be collected. And you may be able to tell that there are numbers on each of the colonies. So software has identified the colonies, and each colony can be tracked over time. And so this allowed thousands of mutagenized colonies to be screened over a time course of several days to derive circadian periods. And the goal here was to find short period mutants, long period mutants, and arrhythmic mutants. And so here you can see a field at a peak time, at a trough time, back at a peak time, and any outliers then were identified by the software. And all of the software was written by Takao Kondo. Again, we're talking now the early 1990s at this point, and just the technologies were not available that we have now. So everything was custom made. And what you can see here is a rhythm of the wild type, also up here. And these are short period mutants and long period mutants, as long as 40 or 60 hours, and as short as 16 or 20 hours. And so we ended up with 17 mutants. We had this range of periods and some arrhythmic colonies. And it turned out that all of these mutants could be complemented by adding one piece of DNA from the genome. So again, that was all done genetically, making a library from of wild type DNA, moving it into the organism, and correcting the circadian phenotype. And so these 3 genes that were identified were named Chi-A, Chi-B, and Chi-C from the Japanese kanji Kai-10, which means cycle. And every one of these mutants had some point mutation that changed an amino acid in one of these 3 genes. These the databases for bioinformatics were not very rich at that time, but they were they did exist, and nothing really could be found that matched any of these genes. And so what happened over the next 15 years really was figuring out, what are these proteins? What do they do? How do they contribute to telling time? And I should say that, yes, if you knock any of these genes out, the cells also go arrhythmic. Okay, so now I'm summarizing for you in one slide what was learned over about 12 years from 12 laboratories. And this was getting from having the gene to having the structures of the proteins and knowing something biochemically about what the proteins do. And so here you see each of those three proteins, Chi-A, Chi-B, and Chi-C. Chi-A is a dimer, Chi-B a tetramer, although I'm going to return to that in a bit. And then Chi-C is a hexamer. And it's a kind of very special hexamer, because when you look at it, you would think maybe that it's two proteins sitting on top of each other. But in fact, each of these monomers is color-coded. This is the N-terminal domain and the C-terminal domain. And but the protein is internally duplicated so that the N-terminal domain and the C-terminal domain are not identical, but they look a lot like each other. And when it hexamerizes, then it gives this structure that looks, we call a double donut, because it looks like one donut stacked on top of the other. Now, when we first had these sequences and had these proteins, the only thing that we could predict was that Chi-C would bind ATP, and it does. And that was because of the only motifs that were present in any of these proteins were a similarity to other proteins that hexamerize and bind ATP in these particular kinds of domains. And it turned out that Chi-C binds ATP, and on only one of the donuts, only on the C-terminal domain, it uses this ATP to phosphorylate two residues, a serine and a threonine. And Chi-A interacts with Chi-C in a way that stimulates this autophosphorylation. And again, I want to emphasize autophosphorylation, the only enzymatic activity that occurs anywhere in this complex is on Chi-C itself. But Chi-A interacts in a way that stimulates Chi-C. Chi-B interacts in a way that blocks the Chi-A stimulation of Chi-C, and then Chi-C dephosphorylates. Remarkably, what I just told you in less than a minute is what tells time in the organism. So that process takes 24 hours. And this is fairly stunning, not only to people who work in circadian rhythms and expect a more complicated clock, but also for biochemists in general, because slow biochemistry is usually not a thing. Biochemical reactions are very fast, and this very simple set of three proteins can tell 24-hour time. And we know that that's true because really stunning work by Takao Kondo's lab in 2005 showed that you can take these three proteins in ATP, put them in a test tube, and you can reconstitute a 24-hour rhythm of phosphorylation of Chi-C. And you can see at the bottom then what that looks like. Let me see, this, the one at the bottom is from my lab. This, what you see at the top is from the original science paper from the Kondo laboratory. But what you're looking at is you can see a non-phosphorylated and a phosphorylated upper band of Chi-C, and you can readily see that just taking samples out of this tube, the phosphorylation rises and falls and rises and falls. And that can be quantified, it can be plotted, you can get circadian periods, and this process actually fulfills all three of those criteria. We can reset it. I don't have time to talk to you about that today, but I can come back to that in the question and answer session if you like. It can reset by environmental cues, and it is, in fact, temperature compensated. So now I'm going to move on to tell you about more recent work. Working with the Kondo and Johnson labs really was a big part of my career in the 1990s, where we established what these proteins are and really we're identifying what the components are that lead to the clock. Not only these three, but also trying to search out and find others that are important for rhythmicity. And then the last 15 to 20 years of my career, I've been working closely with biochemists trying to understand more about what's happening in that test tube. And so the work that I'll tell you about now has been done by in collaboration with Carrie Parch at University of Santa Cruz, UC Santa Cruz, University of California, Santa Cruz, Andy Lee Wong at UC Merced, and my own lab now at UC San Diego. And I'm going to now go through what I already told you, but with a little bit more detail to let you know that we have, and not only our labs, but there are a few other labs that have also contributed to really understanding the biochemistry and the biophysics of what is happening in that test tube and in the cell. And I should say, my lab contributes to the biochemistry. We also do the genetics. So when we identify important interactions that can be seen biochemically, then my lab can make mutations and test those phenotypes in vivo in the cyanobacterium. But we actually know a lot now about how this clock works. I should say these are cartoons, but they are anatomically correct. We actually do have structural information that backs all this up. What we know is that in the morning, or the equivalent of morning in that test tube, Chi-C is not phosphorylated, and there's a very loose association between the two stacked donuts. And again, they're contiguous proteins, but there are interactions between the two rings that are important for function. Chi-A binds to some little C-terminal tails that dangle out, and that stimulates the phosphorylation of Chi-C, first on the threonine and then on the serine. And as it phosphorylates, the protein gets stiffer, and the one ring stacks on the other, and the stacking becomes so tight that there is a binding site for Chi-B that is exposed on this C1 ring. Now, we've also, it turns out that Chi-B is doing something pretty remarkable, and that is that even though several labs got structures of Chi-B that all show it in this tetrameric form, it turns out that this tetrameric Chi-B is inactive. It's an inactive ground state, and that what actually binds to the Chi-C ring is that Chi-B at a slow, steady, and completely in a random manner monomerizes, and those monomers actually unfold and refold into a completely different three-dimensional shape. This three-dimensional shape is unstable and will go back to the ground state unless it's captured really by its binding site on Chi-C. And once it's bound there, it actually forms an entire ring, not depicted here, and that will capture an inactive form of Chi-A. And so at this point now, we have no more stimulation of Chi-C. This stacked form doesn't phosphorylate well anyway, and you end up with a dephosphorylation of Chi-C. And so these processes, where there are many, many small changes that are happening structurally, this is the process that takes 24 hours. And that we can easily assay then by taking samples out of the test tube. And I should say that even though here you're just looking at phosphorylated and non-phosphorylated, you can in fact resolve these different phosphorylation states and determine what's going on during the entirety of the process. Okay, now for working out what I just described to you was done largely using this very simple mixture in a test tube, but that really has a lot of limitations. And among these limitations are that it's very labor intensive, it's low throughput and slow resolution, but there's a bigger limitation. And that is that all this can measure for us is the phosphorylation state of Chi-C. And I started out showing you these beautiful rhythms of gene expression. These three proteins can't control gene expression. None of them is a DNA binding protein. And so in order to see how this timing device which you can think of as the gears of the clock, where are the hands of the clock? How does the information get out? And to do that, we had to knowing how it gets out was really determined genetically. And so we know that we have this dramatic clock-driven gene expression in the organism. Hopefully you're used to looking at heat maps, but what I'm showing you here is work from Aaron O'Shea's lab, where a wild-type culture was entrained to a light-dark cycle and then grown in continuous light. And every 4 hours, a sample was taken, all of the RNA was sequenced, and the levels of each RNA, each transcript from each gene were identified. And now you're looking at a color-coded time series to make this heat map, so that you're looking at expression rising and falling over time in a 24-hour cycle. So even though I had showed you a couple of gene expression from a couple of genes, actually the whole genome is under rhythmic expression. And I had already shown you then that we can take a promoter from one of these genes, hook it up to luciferase, and get these rhythms. And so what we need to do now is we have to reconcile what I told you in that test tube with only 3 proteins. How do we get what I showed you now on this slide, which is the ability to regulate the transcription in the organism, and to do that in 2 major phases. And we actually know from many years of genetic work in my lab and in other labs that and I should say primarily the Kondo Ishiura group, my lab, Carl Johnson's lab, and Aaron O'Shea's lab. From this, we know how the information gets out. And it gets out by the fact that there is a transcription factor called RPAA, whose phosphorylation is controlled by this cyclic process. And the way it's controlled by that is that there are 2 kinases called SAS-A and KIK-A that physically engage with this oscillator. So the oscillator, Chi-A, Chi-B, Chi-C, and then SAS-A and KIK-A early in the day, SAS-A early in the cycle, early in the day in the organism. The SAS-A is physically engages with the oscillator. It binds to the same site that Chi-B does. And when it's on the oscillator, it's a kinase. When KIK-A is off the oscillator, it's a kinase. They both phosphorylate RPAA, and we get this rise during the first part of the day of an active form of this transcription factor. So RPAA will bind DNA only when it's phosphorylated. Later in the cycle, what the events do is they set up a situation where SAS-A is evicted from the complex by Chi-B binding, and Chi-B is able to capture Chi-A, but also to capture KIK-A and turn it on into a phosphatase. So now, at the end of the cycle, we have KIK-A on the oscillator dephosphorylating RPAA. The result of this is that we have a transcription factor that is rising in activity and then until dusk, and then throughout the night, it's falling in activity. And it turns out that the regulation of this one transcription factor is necessary to set up the rhythms all throughout the organism. But so what we'd like to do, we know what the components are. We know that it's a pretty short pathway. And what we wanted to do then was to be able to not just reconstitute the 3-protein oscillator in the test tube, but also to add these output pathways so that we could actually study, mimic rhythmic gene expression in that test tube. But to do that, we needed another assay. And so Andy Lee Wong's lab worked out an assay that would let us look at more than phosphorylation of Chi-C. And the idea here was to put a fluorophore, a fluorescent molecule, on one of the proteins, and we started with Chi-B because it's the smallest and would give us the biggest amplitude of a change. And the change we're looking at here is to take polarized light and shine it into the solution. And if you have a molecule of a protein by itself, it tumbles with fast rotation, and the light that comes out is depolarized. And that's referred to as low anisotropy. But if that protein, Chi-B, becomes part of a bigger complex, it will slow in its rotation, and there will be a polarizing of the light and a high anisotropy. And it turns out that this fluorophore can be put then on other components. So we're no longer with this assay locked into measuring the phosphorylation state of Chi-C. And because this is fluorescence based, it's something that can be monitored in real time. And so this worked beautifully. These are actual data that can be in fact collected in real time of Chi-B in its free state or in its bound state, and you can see we get this lovely high amplitude oscillation. We can, in fact, from the same samples, from partner samples that are being measured for their fluorescence over time, we can also take biochemical samples and look at the phosphorylation of Chi-C, and we could already immediately get information. So, for example, we were able to conclude, and this matched what had been done biochemically and genetically, that the maximal phosphorylation of Chi-C occurs before Chi-B binds the complex. And so this was a remarkably different assay because this can be done in 96 well plates, actually 384 well plates. We can do hundreds or thousands of assays at one time. This allows all kinds of variations, different mutant combinations, and different chemical changes, many, many more things that can be done. And it's not subtle. This is actually, I snapped this picture in December. I was walking through the lab and I saw these data coming off in real time from the instrument and just took a picture of it because it's just so beautiful that just during the assay, you can just go in and look and see what's happening. So really quite remarkable. But the other thing is that we can move that fluorophore around to different components so that not only can we look at Chi-B coming on and off the complex, but we can look at Chi-A, at Chi-B, at SAS-A, at RPAA. So we can look at any of these and ask a different question than where is Chi-C in its phosphorylation state, but also what is each component doing at any given time. And I'll remind you that what we wanted to do was get to the point of mimicking rhythmic transcription. And so we were able to take this one step further, not only to put the output pathways in with the oscillator, but also then to move the fluorophore to the DNA that the transcription factor binds to. So we can do assays where we have an oligonucleotide with the fluorophore, and if RPAA is phosphorylated, it will bind to the DNA and it will have high anisotropy. If RPA is not phosphorylated, it will not bind and will have low anisotropy. And in fact, so at this point, we can measure that. We have a whole in vitro clock. We refer to the 3-protein complex as an in vitro oscillator. That's the gears without the hands. Now we've put the hands on the clock. And what we're doing right now is moving on to actually getting to the point of having transcription occurring in that test tube. How am I doing time-wise? Oh, okay.
Yeah,
So this is actually what I just described to you, this binding of this, we have one phase of information coming out of this oscillator, that's the binding of RPA to DNA. But it turns out that some genes, when RPA binds, it recruits RNA polymerase. And then there's some classes of genes that the binding blocks the association with RNA polymerase. So we end up with this phosphorylated RPA being a repressor of some genes and an activator of others. And that's how we get the 2 phases of gene expression. And that is, in fact, responsible for what you saw in vivo on the right, and from extracts that are taken from living cells. And so the thing that I want to close with is actually an old study that we did with Carl Johnson's lab, and just reminding you that I've talked about things in a test tube, I've talked about things in constant light, but it's a light dark world. And one of the other contributions that we were able to make in collaboration with Johnson Lab is even though everyone in the circadian field has assumed, and it makes a lot of sense, that circadian rhythms have a fitness advantage for the organism, that really hadn't been shown back in the 1990s. It's very hard to show experimentally. And what we were able to do was to use all of these mutant cyanobacteria and asked them to compete and answer the question of, can you see a fitness advantage? And this was really a rather remarkable experiment because we had mutants with different circadian periods. And what you're looking at is an experiment where the cells, either wild type cells or short period mutant, were mixed 50-50 and then grown for a little more than almost 4 weeks in either a short day of 22 hours or a long day of 30 hours. And what you can see is that in the short day, the short period mutant actually took over the population, but in the long day, the wild type took over the population. We could do the same thing with a long period mutant and show that wild type did a little better in the short day, but the long period mutant actually did better in a period that, in a circadian day, a T cycle that matched its circadian period. And then you can see this very remarkably then with the 2 mutants, where it's really clear that circadian rhythms that match the environment improve the fitness in cyanobacteria. Since then, there have been other convincing stories done, other convincing demonstrations using other model systems, using flies, using plants, but this was really a very convincing demonstration early on that what we all expected to be true really can be shown experimentally to be true. And I just like to end by showing the collaborators who were very important in this last part that I showed you of really building out the clock in vitro. The lab of Carrie Parch, her student Jeff Swan, who did a lot of the biophysics, Archana, who was the first to put together the output pathway into that in vitro system. Joel Heisler, who developed the fluorescence anisotropy, Archana and Joel from Andy's lab, and Dustin Ernst and Cheetam Sanchar from my lab, who were doing the genetics in the background to test all of the hypotheses that were coming out of the in vitro work. And then Ming-Shu Fong from my group, who has really become the master of the in vitro clock. And very recently, he's been able to demonstrate actual rhythmic transcription in a test tube. And I'd also like to end then by remembering to Cal Kondo, who contributed so richly to this field. We lost him in November, and he will be sorely missed. And with that, I'll be happy to take your questions and leave you with this QR code, because one of the other things that my group has done, not my group, that I have done wearing another hat, is I was funded by the Howard Hughes Medical Institute to teach a course for 5 years in which we made videos in the field of circadian biology. These are all online. They are they're freely available, and we encourage you to go. They're not just about cyanobacteria. There are 3 about cyanobacteria, but there are many that are about humans and health and lots of different topics in circadian biology. And with that, I'll close and take your questions.
Wow. Thank you very much, Susan. That was a really nice talk. What a great collection of research.